Hi, I'm Alyssa Long, and I'm a research scientist in the Caspary lab at Emory University. In this short talk, I'm going to show you some highlights from our recent research, which has been published in the Journal of Cell Science. Our lab has long studied a protein called ARL13B, which normally lives in tiny hair-like structures called cilia located on the cell surface. Mice that completely lack ARL13B die before birth, so it has been hard to determine what the protein does throughout life. A common technique to get around this problem is conditional deletion. This method starts with a normal mouse and removes a gene and the protein it encodes in a specific tissue or at a specific time. However, this method can be somewhat limited. You have to choose which tissue or time point to study. We chose a different approach. We conditionally expressed an engineered fluorescently tagged version of ARL13B called ARL13B-Cerulean, using a mouse model developed by the Mort and Mill labs in the UK. With this glowing ARL13B turned on everywhere in animals that were otherwise missing the protein, the mice survived to adulthood and they looked healthy. They moved normally and they had no obvious problems in their kidneys, pancreases, or brains. We were surprised to discover one major exception. Rescued males were infertile. Although their testes were the normal size, they produced very few mature sperm, and the sperm they did make often had misshapen heads and short abnormal tails. The sperm's tail, which is also called a flagellum, is like the cilia I mentioned before, where ARL13B lives. The flagellum is critical for sperm movement, and the sperm we isolated had movement defects consistent with reduced fertility. Taken together, our results show that ARL13B-Cerulean can replace the normal protein to support development, and that this mouse line can be used as a flexible tool to study cilia and ARL13B function in many tissues. We also revealed an important new role for ARL13B in spermatogenesis and the possibility that defects in this protein could contribute to male infertility. We're currently looking at the function of ARL13B in spermatogenesis. What partners does it work with? Does it play a direct role in sperm cells, or does it have an indirect effect? And does it work differently in cells that have cilia versus flagella? To learn more about our work, check out our full manuscript, available on the Journal of Cell Science website. Thank you.